She has also been with us previously as a speaker on the same topic, and her session was truly insightful and well appreciated. So let's warmly welcome Dr. Awani to the session webinar topic.
Thank you so much, Dr. Rohan, for that great introduction. It feels, very awkward that I still feel that I am a student myself. Just passed out from MD from SMIMER Medical College, and I still feel like I'm still a student. So getting introduced by you, it means a lot. So thank you so much.
Thank you.
A very good afternoon, everyone who has joined us for this session. Second round of Simplified Bioassays. It's great that the first round was very helpful for all that we're back with this session. And first and foremost, I request everyone who is joined to please mute yourself so that we can have a really good flow of this session. One thing that I would like to mention is when post MD, when you start your life, either it's in academics or industry, anywhere, something that we need to learn about is work-life balance and how to juggle everything. That is something I feel it's called adulthood. I was not expecting to travel today, but then at the last minute I had to travel, and I'm taking this session from the airport. So please let me know if there is any disturbance mid-session or you are unable to hear me, so that I can fix the issue. Otherwise, we're set to go. I'll share my screen and we'll start the session.
Yes, we can start the session now.
Okay, is my screen visible to everyone?
Yes.
All right. So I'll start with my session. Once again, a very good afternoon, everyone who has taken time out and joined here today. I am Dr. Awani Shah, working as a regional medical advisor in oncology in Mumbai at Dr. Reddy's laboratories. But before starting my session, I would like to give a quick disclaimer that I'm a full-time employee of Dr. Reddy's laboratories, but the session is purely educational purposes, and views expressed here are personal views and do not necessarily represent views of Dr. Reddy's laboratories. With that, start with my session with first and foremost words of wisdom. I am a huge MS Dhoni fan, purely because whenever I watch his interviews, there are always some good takeaways that I can carry along with me in my personal life or in work, and not necessarily that what he says is meant for sports only, but to also imbibe personally and professionally as well. So with that, I always start with words of wisdom from MS Dhoni. First and foremost, so I would believe that everyone who has joined here to this afternoon would be from the first year junior residency to second year to perhaps final year junior residency. But all I want to tell all of you is that do believe that I believe in giving more than 100% on field, and I don't really worry about the results if there is great commitment on the field. That's victory for me. So when it comes to bioassay, just give your 100% bioassay, but throughout your MD journey, give your 100% without worrying about the results, because if you've given your 100%, the results will automatically show in a positive manner. And the second most important thing that I like what MS Dhoni has repeatedly mentioned, that the process is more important than the results. Follow the basics. If you take care of the process, you will get the results. So trying to understand bioassay, let's start with the basics. Understand the basic fundamental, not just in bio, but anything else. Understand the basic fundamental, and then understand it in more depth. So with today's presentation, I would like to give you an overview. My main focus would like to be in the practical part, but unless and until you don't know the basic theoretical part, it would be difficult to understand the practical session. But let me tell you one thing also, bioassay is extremely vast. The first session I conducted on simplifying bioassay, we kept it for 1 hour. And I realized, okay, 1 hour is not enough. So then this time I asked Dr. Rohan, so let's keep it for 1.5 hours so that I can try to cover as much as I can. But
Sure.
even in 1.5 hours, I cannot, complete the entire bioassay. It's extremely vast. It usually would take multiple sessions in this. So I'll do my best to, cover the basic theoretical part so that you can understand the basic terminologies. But I urge all of you to read, post this session, go back and read so that you can get a better grip with it. And at the same time, once I complete the theoretical part with introduction till the type of tissues, I'll quickly also cover the practical part so that you can understand how to present it during your exams or in your daily activity of MD curriculum. And most importantly, how do you present the bioassay practically, right? So with that, I'll start my session. I urge everyone to keep their mics muted, and let's park the questions till the end if time permits, or please write it down in the chat box. If at the end of the session time permits, I'll try to answer as many as I can. Otherwise, I'll request the organizers to send me those questions, and I can try to later on reply, as I also have a flight to catch at 4. So without much delay, let's start with this session. So as you can take a look at this image depicted on your screen. So this slide highlights branding and packaging consistency of a cough syrup. When you see it visually to your left and to your right, the visual design, the labeling, the packaging, everything is same, right? Everything is same. But however, this bottle, which represents of a cough syrup, is used in the two different ones, according to the color. They're used for a different type of cough syrup. So while one is used in particular for dry cough, or as, for example, non-productive cough, another is used for wet cough, productive cough, or for mucus. So my key question here is, how was it known that a particular drug used in this cough syrup will suppress the cough, or it will loosen up the mucus and be used as expectorant cough syrup? So how was this known? Well, a simple answer to this is by doing an assay. So an assay is the determination of the amount or biological activity of an active ingredient present in a unit quantity of pharmaceutical preparation. Now, in simple terms, it tells us how active the drug actually is in the product. So, for example, there are 3 types of assay. So one is physiochemical assay, second is immunological assay, and third is biological assay. So when you look at physiological, chemical, or psychochemical assay, one of the most common example of it is chromatography. Now this measures the chemical property of the drug. Another type of assay is immunological assay. So this uses antigen-antibody reaction to detect and quantify substance. And the third most common type of assay is biological assay. So where it measures the drug's effect on a living cell or organism. So together, these methods ensure drug quality, potency, and safety. So with this, coming to the topic that we are all joined in today is bioassay. Now, let me once again tell you that, I'm just talking about the conventional type of bioassay, and, the traditional method of doing bioassay is what I'll briefly discuss in this session. I will not be going much into detail regarding the simulation model or the calculation part, because we don't have enough time for that. But coming back to bioassay, what is bioassay? This is the most common Viva question that you should know, as well as a short note question that would be asked in your examination. So when you break down bioassay, the most common line a Shakespeare has said, what is in the name? Well, here name depicts everything. It's, at least in science, it depicts everything. So bioassay, when you break down this word into two, bio and assay. So bio means living material and assay means assessment at a laboratory. So bioassay is defined as a comparative assessment of potency of a test compound, which is denoted as T, to the standard compound, which is denoted as S, on any living animal or biological tissue. Now, it's primarily used for determining the quantitative relationship between the concentration, which is the dose, and the magnitude of response. Now, most common type of methodology used in bioassays either in vivo, in vitro, or in silico. Now, these again, I'll tell you, is Viva question. In vivo, what is in vivo, what is in vitro, and what's in silico? So to briefly tell you about it, in vivo, in pharmacology, it is the experimental process which is done outside the living body using isolated tissue preparation, isolated cells, and enzymes. So that is called in vivo. That is called in vitro. So again, I'll just repeat, in vitro. In pharmacology, it is the experimental process which is done outside the living body using isolated tissue preparation, isolated cells, or enzymes. So in vitro, so that is called as in vitro. Now coming to in vivo. So in vivo, it is the experimental process which is mainly done inside the living body. For example, pharmacodynamic study, pharmacokinetic studies, these are done inside the living body. Hence, it's known as in vivo. And third type of bioassay is also called as in silico, which all of you would, or majority of you would now be using in your MD exams or in your practical examination. So in silico, meaning it is the experimental process which is performed on computer or via computer simulation. Right. So these are three different types of bioassay: in vivo, in vitro, and in silico. Now, two more types are there: in ex vivo and in situ. Now, I would like to leave it up to you guys to, after this session, go on and find out what is ex vivo and what is in situ. But please remember what is in vivo, in vitro, and in silico. Moving forward, understanding bioassay and all this is fine. But we should and we must acknowledge those scientists who has helped us in discovering this. And with that, our first and foremost is Paul Ehrlich. He founded the standardization of diphtheria antitoxin. He contributed significantly on the work of immunity, as well as pioneered the technique of chemotherapy in medicine. And it's not just Paul Ehrlich, but also Rudolf Magnus. He was the first scientist to design the arrangements of bark for excised organs, especially for intestinal strips. And this was as early as 1904. So he designed the assembly for recording the contraction of isolated tissue. As we all know, we've seen the organ bath. It's also called Rudolf Magnus organ bath. So this is the scientist, who invented this, who designed this assembly, where now we can perform the bioassay experiment. And along with this too, there is also John William Trevin, who designed the concept of LD50. LD50 meaning lethal dose 50, as well as first person to design the bioassay of digitalis. Along with this, Alfred Joseph Clark, the scientist who invented the drug receptor theory and the dose response curve agonist antagonist receptor interactions. So it could, bioassay could not have been possible or for us to even learn without the contribution of this great scientist, to which we must acknowledge them before, starting our journey in understanding this bioassay in today's session. So with this, I come to the most important part of bioassay, which is known as the principles of bioassay. So please note down that this can be a highly possible viva question, but also a question that can come into your examination, your final exams or your prelims or any internal exams. So the basic principle of bioassay is to compare the biological effects of a test substance with the international standard preparation of the same compound. Now, thereafter, it is determined how much the test substance is required to produce the same effect as produced by the standard. So these are the four most important principles which I have just broken down into bullet points for you all to understand and try to remember them, because so that you can write down these points. These are the key points that you can write down as well as highlight when asked during your viva examination. At the same time, now we understood what is bioassay, what is its principle, but where is its application? How do we use bioassay? So bioassay is the only method of assay used if there are certain criteria where it can be used, or where it's the only method that can be used. One, when the active principle of drug is unknown or cannot be isolated, for example, insulin. Second, when why bioassay is important and where we can apply is when the chemical method is either not available, or if it's available, it's too complex and insensitive or requires high dose. Again, insulin, acetylcholine. These are some substance which requires very high dose. Another application is chemical when the chemical composition is not known, or when the chemical composition of the drug is different but have the same pharmacological actions and vice versa. For example, cardiac glycosides or catecholamines. So these are some of the applications where it's important to use bioassay, or perhaps it's the only method that we can use, in the experiment. So again, a highly important thing for you to know, can be asked in viva. Definitely, it will be asked in viva, as well as in your theory examination. Now, for any essay that we do, there are certain characteristics, but certain characteristic to say how or why is it a good essay. So there are few points that you need to remember when performing any essay, or suppose even bioassay. Sensitivity, how sensitive it is, how specific it is it reproducible, is it accurate, and is it precise? So when I say sensitive, it means that minimum amount of drug which is able to produce a biological response. Now, the tissue which responds to minimum amount of drug is said to be the most sensitive to a particular drug. When I say specific, what do I mean by that? That means the ability of the analytical method to determine only the component that we wish to determine, right? Without determining any other component. So it's very specific. If I'm trying to determine something of X, then the result should give me of X only and not of Y. Third point is reproducibility. Now, repetitive and same results obtained with the same process, method, materials, but in different labs, under different conditions, under different person, under different environment, under different apparatures. Will this still produce or reproduce same or in a similar manner? That is a sign of a good characteristic of an essay. Third thing, or the fourth thing, accurate. How accurate? The degree of trueness which is measured or calculated to its true value. And precise, precision. Precision is very important. The degree of exactness of a calculated value falling exactly within the range. So accurate, we say the degree of trueness. Precision or precise, the degree of exactness. So these are 5 characteristics of a good assay. Now coming to bioassay and its type. So there are different types of bioassay. Now, bioassay most broadly is divided into quantal assay or graded assay or direct endpoint assay. Okay. Now, quantal response, the response is in the form of all or none. Either there is response or either there is no response. There is no in between. So either something is black or something is white. There is no gray in it, right? So that is called as quantal response, all or non-response. Now, the drugs producing quantal effect can be bioassayed by endpoint methods. And the threshold dose producing a predetermined effect is measured here. Okay, and the comparison between the result of standard and a test. Now, example I would say is, bioassay of digitalis in cats, insulin-induced hypoglycemic convulsions in rat, determination of LD50. So these are, will give you a basic yes or no answer, all or non response. Now, with graded assay, it is again divided into different types of graded assay. Now, graded assay, when I say that, it means that the response is proportional to the dose, and response may lie between no response and maximum response. So the types can be bracketing, matching, interpolation, multiple point. Now, multiple point graded bioassay is again divided into 3 point, 4 point, 6 point, and 8 point. So these are different types, and I'll try to just highlight the basic points of each type of this bioassay as we move further. But one thing I want you all to remember, that in all bioassays, a reference standard is required for you to compare it, right? So S, as I had mentioned in the definition also, standard concentration versus the concentration which you're going to test. So S and T. So with this, I would like to now move on to the dose response curve. Now, you've all heard of DRC, dose response curve. So it stands for the dose response curve, which represents a simple X and Y graph, a X and a Y graph. It represents a simple X and Y graph relating to the amount of drug to the response of the receptor. Now, DRC demonstrates graded response to drugs or agonists where there is an increase in response. When an increase in response is recorded with subsequent increase in the dose or the concentration of the drug. Now, the typical DRC, as you can see, a typical DRC is sigmoid shape, or also known as S shape. All right. So you can see the B graph in the diagram to your left represents a regular DRC, dose response curve. It is sigmoid curve, and its wide range of doses can be plotted. That is one of its advantage. Now, what does this DRC indicate? So there are 4 points that I would, or actually 3 main points which I like to, highlight, that the importance of having a dose response curve. One, that the relative potency of the drug or agonist when the curve is measured here. So when the curve is more towards the left, it indicates that the drug is more potent. I would like to highlight again that you must be aware of the terminologies. What is potency? What is efficacy? When I say that it's more potent, what does that mean? When I say something is more efficacious, what does that mean? So, I urge all of you to go back and read after this session right from the beginning, so that you can understand. As I mentioned that, until and unless you don't know your basics, it will be difficult for you to understand further on. So potency, efficacy, the slope of the curve, these are very important terminologies related to bioassay and DRC. So you must be very clear with its definition. So the first, I said, importance of DRC, what it indicates. First, it indicates the relative potency of the drug. The curve more towards the left, it indicates that the drug is more potent. Second, the error and reliability. In this, the precision, it is indicated by the slope of the curve. So the steeper the curve, the more precise the assay is, and vice versa. Now, the third thing that DRC indicates, it's the efficacy of the drug. So it's indicated by maximum response of the curve. The higher the maximal response, the higher the efficacy. So these three things, you need to know what is the importance of DRC and what it indicates. So I mentioned the potency, the efficacy, and the slope of the curve. Now, second thing that I want to mention, the different parameters which are characterized in a dose response curve. So as you can see it, the diagram to the right, the C, you can see this different type of terminologies here. So submaximal effect, maximal effect, supramaximal effect, ceiling effect, right? So what is the different parameters which are characterized by dose response curve? The first and the foremost is maximal or ceiling effect. Now please note this down. If I am emphasizing on certain terminologies, that means they are likely to be asked in your Viva exam, or in your not just actually in your Viva exam, but also when you write a short note or theory based question, that you'll be writing down this terminologies also, or you'll be drawing down this graph also. So it's important for you to first understand this terminology. So when I say maximal or ceiling effect, it means that it's commonly referred to Emax or efficacy. So the maximum effect is different from potency. Different efficacy suggests different mechanism of drug. Second thing is positioning of the curve. It gives you the idea, about the affinity or potency or ED50 of the drug. Now, if the curve is, as I mentioned before also, if the curve is more towards the left, it's having lesser ED50 and it's more potent. So that's why again I'm emphasizing, you need to know the terminology, potency and efficacy, and what it means. Also, again, the slope of the curve, as I mentioned, the slope of the curve will determine not just the potency but also the precision. So these are the characteristic of a dose response curve. Now, coming to most important part, as you can see here, log dose or log DRC. When we are plotting DRC, the advantage is wide range of doses can be plotted. And what is that called? That is called the arithmetic addition. So in arithmetic addition, the doses are added in arithmetic progression, like 1, 2, 3, 4, 5, 6. But when you plot in the log DRC, it is called geometric addition. So in geometric addition, it's like 1, 2, 4, 8, 16, 32. So in which manner the doses are added or plotted in DRC? For the convenience of plotting a DRC, the doses are increased in geometric progression, called as logarithm intervals. So this log DRC, where the importance or the advantage of using a log DRC over a DRC, that the biological responses are linear to the log dose. Therefore, the progression in response during a geometric addition is near linear, and you can more doses range may be covered, but using a minimal or a lesser number of the dose. So when you see this log dose graph, it is rectangular or also called as rectangular hyperbola, whereas a normal DRC is a sigmoid curve. While wide range of dose can be plotted, but when you see a log dose graph and then those tiny squares, it is difficult for you to plot ranging from 1 to 20 or 40 or 50. And that's why a geometric addition, so 2, 4, 8, 16 is used in log dose response curve. So again, this is a Viva question. The advantages of log dose, the disadvantages of log dose, or why is log dose preferred over DRC, the shape of it, the advantage, and how can you interpret them, as well as the different terminologies in a log dose response curve. The submaximal effect, maximal effect, supramaximal or ceiling effect, and most importantly, we use a log dose response curve. Why? Because the dose should be selected in a linear region due to better dose discrimination. So as you can see, from 25% to 75%, this is where the dose should be selected. Again, this is a Viva question, and it can be and it will be asked to you. Once you plot a log dose response curve, in which part or which section should you select the dose? So it should be ideally selected between 25 to 75%. Why? Because in this region there is linear consistency in the graph. So better dose discrimination is there. Again, you'll understand it better when you understand such terminologies. But owing to time, I have to move quickly from the theoretical portion. So moving on now to direct endpoint assay. So why is direct endpoint assay used? Its aim is to determine the threshold dose required for a response. Now, its principle is to measure direct response of dose of standard, S, and test. Now, the ratio between these doses estimates the potency of the T preparation relative to S. And criteria used for direct endpoint assay, that the response should be clear and recognizable. The most common example is assay of digitalis in cats. So the advantage of using direct endpoint assay, the drug effects appear rapidly and they are easily recognized. The drug effect is directly proportional to the drug dose and rapid endpoint detection. So while there is advantage, there is also disadvantage. You must agree, right? In anything that we have, there are always two sides of the coin. So while there is an advantage of direct endpoint assay, there is also a disadvantage that only toxicity or high dose study is possible. And dose ranging studies cannot be done. So, each type of assay has an advantage and a disadvantage, and it's used accordingly. When we come to quantile assay, as I mentioned before also, that the doses of standard and test are compared for all or non-response. Either there is no response or there is a maximal response or a maximum response. And this is usually done for drugs producing response that cannot be quantified or recorded. So, for example, insulin-induced hypoglycemia conversions in mice. So either there is conversion or there's no conversion. Cardiac arrest in digitalis. So either there is cardiac arrest or there is no cardiac arrest. Determining LD50, the lethal dose. So either the patient or the participant or the subject died or it's alive. There is no in between. So that is what we call quantile assay. Moving on to graded assay. And again, I told you there are different types of graded assay. So for you to remember what is graded assay, it's the response obtained by administering increasing amount of drug to a single subject or to an isolated tissue. Now, this explains ideal quantitative relationship between dosage and response. And there are 4 types of it: matching, bracketing, interpolation, and multiple point. Now, again, multiple point are of different types. There can be 3-point assay, 4-point assay, 6-point assay, and 8-point assay. Now, before I move on, if we have first-year juniors or first-year PGs joined in, or we have second year PGs who have joined in but not yet started with bioassay. This may feel overwhelming, but trust me when I tell you that, once you at least listen to my session, get the basic orientation of it, and then go back and start reading it, you will get a better understanding, and you will realize now of, what I meant, or what I mentioned. You'll try to recollect it, and then you'll get a better understanding of it. So that's why I feel that, with bioassay, when we are not performing in particular, when we don't see something visually, it's hard to understand. Or even when we're reading for the first time without someone explaining it to us, it's very difficult to grasp, to understand. So that's why I felt that, I want to simplify this bioassay for everyone and for all my postgraduates to make their life simpler. And trust me, right now, if you feel this is overwhelming, that's completely fine. It's very natural to feel that way. But once you go back and read, and I really hope that you read, you'll get a better clarity with this. So with this, we move on to matching bioassay. Now, matching means, as the name suggests, right? The standard and unknown are matched by a trial and error process. I have bolded this, a trial and error, because it's a peculiar terminology used when it comes to matching. So if an examiner asks you about matching and you don't mention a trial and error process, then the marks will be deducted, because this is something very in particular for matching. So again, matching is the standard and unknown. The standard and the unknown are matched by a trial and error process until they produce equal effect. Now, response is matched at only one dose, so does not need dose response curve. So as you can see here, this is the standard. Now, any of the standard is taken. Here they have taken this S1 with a known concentration. Now, that same concentration of standard is matched with the test. So they are trying to match, So this response, it's matching here. This is where they get to know that the response of test is matched with the response of standard, either S1 or S2. So this is basically very simple, and that's what we call matching. And here we don't require a dose response curve. So the advantage of matching is it's very simple and sample size is very small. The disadvantage, on the other hand, as I mentioned, everything has advantage as well as disadvantage. So here the disadvantage is that it does not involve the recording or the concentration, the recording of concentration response curve or dose response curve. Therefore, there is no sign of parallelism. So sensitivity of the preparation or the procedure is not taken into the consideration. It's not accurate. Precision and reliability is not very good. So as I mentioned, those 5 characteristics to call it a good assay, they must be present. But when you're doing matching, there is no precision, there is no accuracy, there is no reliability. So that's when, the disadvantage comes in with this type of assay. The example of matching assay is, bioassay of acetylcholine in frog rectus abdominis muscle, the simplest method that can be done. Next comes bracketing assay. So the test response is bracketed between 2 responses. Again, as the name suggests, bracketing. You are bracketing between 2 responses, one higher and one lower of the standard drug. Now, the potency of the test drug is directly calculated from concentration of standard drug or by interpolation through dose response curve. So you can see the dose response curve of standard, and then the test response is bracketed between 2 doses of standard. So there are S2, S1 bracketing the test response, and then S2, and then they will continue with another S1. Or here, S2, you're bracketing T, and then again S1, S2. So that's what it means, that you're bracketing the response between you're bracketing the test response between 2 response of the standard of the drug. Now, the advantage of bracket assay, it's preferred test sample volume or the preferred test sample volume is too small, and it's the simplest way of graded response assay. But on the other hand, the disadvantage, there is poor precision, poor reliability, and unable to calculate error. I still remember this when I was giving my final MD exam, and during the Viva, one of the external examiner asked me, which type of essay will you consider if you want to know the precision of the response? Then which type of essay would you consider? So I leave this question up to you for you to go back, read, and then try to answer this question. But I clearly remember that I was asked this by the external examiner. Next, we come to interpolation. So interpolation, as the name suggests, again, it is to calculate the concentration of unknown from the known. And a log dose response curve here is required, and it's plotted with the standard on a semi-log paper. So remember, when you are trying to plot a log dose response, it's plotted on a semi-log paper. You need to know the advantages of this, the disadvantages. Everything is advantage, everything is a disadvantage, right? And these are the points where Viva questions will arise. And also one thing, remember, whatever you answer, your next Viva question will lead from that. So remember to, if you are confident about the answer, if you are confident about knowing something and you are answering, be prepared that your next question will lead on from what you have answered. So please do not try to bluff, because the examiners, they are very experienced, they are very smart. They know if you know your concept or if you don't know the concept from the way you answer, right? So back to interpolation. So here, the log dose response curve is plotted with the standard on a semi-log paper. Concentration, the response is then interpolated with this log dose, and then they try to match. Here to know the unknown concentration from the known concentration. So the known concentration is plotted, a linear line is drawn, and then whatever log dose they're trying to know, the unknown concentration, those responses are matched. So the responses are plotted in the y-axis, right? And then accordingly, they can match whatever they require. So standard and the test or the unknown. So advantage, less time consuming, and it's reliable. Now moving on to multiple point assay. So just give me 1 minute. Okay, so it's 2:40. And we still have to get to the practical part, but we're nearing the end of near the theory part. Okay, so multiple point type of bioassay, repeated responses are recorded. It minimizes the tissue sensitivity error. There is decrease in methodological errors. These are the advantages of multiple point. Now in multiple point, it involves the selection of one, which is known as 3 point, or more, 4, 6, 8 point responses of the test. Now the compound of the test compound, and it's compared with 2 or more responses of the standard. So when you are trying to do a 3 point assay, the dose of the test is 1, and you are comparing with 2 standard doses. So the response will be 9. I will explain to you in the next slide how such responses derived. If it's 4 point, that means you are going to have 2 tests and 2 standard. So it will give you 16 responses. When it's 6 point, as explanatory, or as you can see on screen, 3 dose of the test, 3 standard, 36 responses. 8 point, 4, 4, and 64 responses. So now, whichever you select, suppose it's 3 point, 4 point, we'll go with the standard 4 point. 2 tests and 2 doses are selected. How will you get 16 responses, and how will you decide the responses? Now that is decided by Lapping square randomization. Please note it down, a Viva question and also a theory question. Okay, definitely a Viva question. So Latin square randomization, it's designed in an experimental design that it can be used to control the random variations of two factors. Now the design is arranged with an equal number of rows and columns, right? As you can see, equal number of rows and equal number of columns, so that all combinations of possible values for the two variables can be tested multiple times. Now, this design is used to reduce the effect of random factor, and that is the advantage of Latin square randomization. So here I'm trying to show you a 4-point essay. So with a 4-point essay, as I had mentioned, 2 responses of standard are taken, S1, S2, and 2 of test are selected, denoted as T1, T2, from the dose response curve for the consecutive 16 responses of Latin square. So 4 by 4, 16 responses. Now the question arises, how will you randomize this? So it's very simple. You, in your first row, you write down S1, S2, T1, and T2. In your second row, then you start with S2. So S2, T1, T2, you bring this over here to the end and it's S1. Then in the third line, now you start with T1. So T1, T2, back to S1, S2. And then in the fourth line, you start with T2. So T2, S1, S2, T1. So this is how randomization is done. Now, whether it's whether it's 3 point, 6 point, 4 point, any, but this is how randomization is done. So you first write down S1, S2, T1, T2, or suppose it's S1, T1, T2, and then this is how you start doing your randomization. The importance of doing a Latin square randomization. It's that the sensitivity of the tissue varies with time during the course of experiment. So by doing this randomization, the response of the dose is affected by the size of previous dose. So the dose should also be evenly distributed throughout the assay and randomized. You don't want to have any mix-up that you used S1 and then the next response that you're trying to do or try to test, you'll again use S1. No, we want to have a clarity of what we're doing in the sequence, so that everything is equally distributed and randomized, but not at the same time, right? So this is Latin square randomization. Please note it down that you will definitely be asked, or at least to know the concept of it. So as you can see, this is just one of the examples which I had taken, that the contraction. So the contraction which is measured in mean and the dose which has been taken. So this is how they do the randomization. So the first randomization they took S1, then the second time when they did S2, they took S1. What was the dose taken? So this is how randomly it has been selected and constructed to give you those 16 responses. So this is a 4-point essay giving you 16 responses, and how it's measured to get your S2 divided by S1 equals T2 divided by T1. So again, I'm not going into calculation because this is another session on calculation in particular to understand the mathematical part. But right now I just want to focus on the basic theoretical part so that you can get a basic orientation. So with this, I complete the theoretical portion of bioassay. Now we come to the practical portion of bioassay, with having precisely 36 minutes left, right? So before going into the practical part, we always need to understand the assembly of bioassay and bioassay apparatus setup. So four most important points here, and I'll tell you, with everything moving onwards now, every point there would be a viva question. If there are very experienced examiners who are coming in your during your exams, who has done the traditional method of bioassay when they were doing their MD or pursuing their MD, they would ask you questions left, right, and center so that you know what you are working with. So first and foremost, the common things that you need to know in a bioassay apparatus setup: the rotating drum and thermograph, the outer bath and inner organ bath, tissue holder, oxygen supply, fulcrum, liver, heating coil, thermostat, stirrer. Now I'll show you a video of the student organ bath, its parts, its arrangement, so you get a basic orientation of how it's set up, as well as even give you the importance of those essential parts of the organ bath. So now, before going into this, let me share another screen to show you the video of this. Okay, Dr. Rohan, can you confirm that this is visible? Another screen I put up.
Yes, Dr. Awani, it is visible.
Just one more minute. Just give me 2 minutes. I'll stop sharing this screen and I'll share the screen again with. Okay. Dr. Rohan, can you please confirm my screen is visible with the student organ bath?
Yes, the organ bath is visible.
All right. So everyone who has joined, I'll play this, and I want you to see. So this is the student organ parts and its arrangement. So first and foremost, stand of the organ bath, the organ bath chamber, with this, you see the water inlet and outlet along with the clamp. The water filled in the organ bath chamber. I'll explain this, once you see the entire video of the assembly and its parts. So within this organ bath chamber, there is water which is heated up to 37 degrees Celsius. This organ bath also contains a thermostat. Now, each of this assembly is very important to understand why is it there, what is its important, and many viva questions can come directly once, in your practical exam, the examiner can also ask you to present or explain the organ bath setup. There is also heater in the organ bath chamber. Within this organ bath chamber, there is a clamp holder which will hold the stirrer. Another Viva question, the importance of the stirrer, the importance of the thermostat, the heater, why is the water, temperature to 37 degrees Celsius. Another apparatus in this organ bath chamber. It's called the organ tube. So please remember, the organ tube and the organ bath chamber. So it can be outer organ bath, inner organ bath. There are various names to it. So inner organ bath is the organ tube. In this organ tube is where there is an aeration tube. In this aeration tube, as you can see here, is where you'll mount the tissue. Now, in this organ bath, there will also be a fulcrum, a writing lever. So simple and frontal one. Now you can have theory question as well as Viva question with types of lever, different types of lever. Its importance in the sense of how it is made. Okay, this is a writing lever, simple and frontal, which has load in the back. Why is it important to have the load? What will, why its placement, the required placement for its magnification. Now, as you can see in this writing lever, there is a tissue connected to the aeration tube, and that tissue is also tied to this writing lever. There is a gas coil inside the organ bath chamber. To this glass coil is attached the physiological salt solution. This glass coil is also attached to the organ tube. So each and everything is important, and you'll be asked why. What's the importance of the stirrer? So now with this writing lever, that is again attached to this chymograph drum. This drum also has an assembly called the electrical assemblies, where you can start the drum or stop the drum or adjust the speed. RPM is the speed. This is the chymograph drum with the chymograph paper. Now, types of paper, different types of paper. How do you mount the paper? How do you mount the paper, take the response? So all this is very important for you to understand, because all these are Viva questions. And if you don't perform it, but at least you see this, you will get the basic idea of it. So now that I've shown you the video of it, I'll share my screen again, get back to the presentation, and now try to explain you. So, Dr. Rohan, is my screen visible?
Yes.
Okay. So with this student organ bath, the parts and the arrangements, the video that you saw, now there each assembly has an essential part. So the first thing, here, the rotating drum and chymograph. So this rotating drum, it's also known. So this rotating drum, it's also known as Sherrington rotating drum, and its speed can be adjusted by attached gear and lever. So there's a lever here, there's a gear there, gear with clutch, as you can see. So the speed can be adjusted. Now, it's important, the type of tissue that you use, and the speed that you choose. What it's important to know why or which type of tissue you are selecting in terms of slow contractile tissue or fast contractile tissue. Because for the slow contractile tissue, the drum is kept on a low speed, and for the fast contractile tissue, the drum speed should be kept comparable high speed. Now, the standard speed of the drum is 1 revolution per 96 minutes, and this is another Viva question that would be asked to you. Now, on this kymograph drum is the kymograph paper. So, kymograph is the paper used to record the response of the tissue or the muscle against the known or unknown concentration of the drug. Now, that paper, if you've seen it physically, it has two sides. One is the glossy side or the smooth side, and the other is the rough. So the examiner will ask you, how will you mount this paper? So while mounting the paper on the drum, one should make sure that the smooth surface is outside. Why? Because it helps and it doesn't produce resistance in the movement of the liver on kymograph. And the rough surface is placed inside. Why? Because it helps sticking the paper on the drum. Now, please, everyone, I would like to, tell you that whatever I say now, anything onwards from here, everything can be a Viva question. Now, the outer bath or the outer water jacket. So this is mainly used to store water outside the inner organ bath to maintain the temperature for experiment. It is made up of perspex glass. Then we have an inner organ bath. So it is transparent and it's made up of glass to absorb the tissue during experiment. The inner bath has varying capacity from 5 to 50 ml. Proper marking, so there are proper marking done inside this tube to observe the tissue during the experiment. And the inner bath has a varying capacity of 5 to 15 ml. And also because of this marking, it helps to fill the physiological salt solution also. There is a fixed level every time that, when you fill the physiological salt solution inside this organ tube or the inner tube. And why do we put physiological salt solution? Because you are mounting the tissue and keeping it inside here. Intact tissue is, kept inside here. Now there is a tissue holder. Along with it, it's also the oxygen supply. So tissue is attached here and inside the inner organ bath with the help of a tissue holder, and it also supports the air or oxygen supply through its motor. There is fulcrum here. The writing lever is attached to this and helps free vertical movement. So free vertical movement to record the response of the tissue against the drug. We have heating coil here. So heating, as you can see, the thermostat, the heater. So heating coil and thermostat, heating coil is attached to the outer bath and it's made up of iron. It maintains the temperature outside the inner organ bath as well as the physiological salt solution which moves in the inner bath through the coiled inlet. So you can see there would be a glass coil, there is a coiled inlet. Whereas most thermostat, as the name suggests, it maintains the static or the constant temperature throughout the experiment and it avoids variation in the temperature. I remember, so Dr. Panwar, he had asked us, suppose this we are running through electricity, but suppose, there is no electricity or during the time of experiment, the lights go off, then how will you maintain the temperature? How? Because the thermostat won't work without electricity. So how will you maintain the temperature in the organ bath? So, such kind of questions can ask when their experienced examiners who has done personally such kind of experiment can ask you. So in such case, if the power goes out and there's no electricity, then you boil the water on gas or something, and then you try to maintain the temperature. Putting the thermometer inside and measuring the temperature. Then, as you can see, stirrer. So it gives circulating water to maintain the similar temperature throughout the outer organ bath. So, once the water is added, but it needs to be circulated to maintain the temperature, to distribute the temperature evenly. Therefore, the importance of stirrer. Now, fixing of recording. So, once you have recorded the response in this chymograph paper, you take it out. Now, how do you fix it? So, using a fixing solution, which is also called as shellac. So, fixing solution is shellac and phosphorus is saturated in alcohol. Then comes to the writing lever. So, as I said, they have used a simple frontal lever. There are many different types of levers. So, please go ahead and read it in the book to understand their importance of it. Now, lever is a mechanical instrument which is based on the principle of static equilibrium. So it can be of type 1, type 2, type 3. Now there is peculiarity with each type of lever. So type 1, the fulcrum is located between the input effort and the output load. So this is the load, output load, input effort. For example, like in Bioassay, or I'm sure you've seen a seesaw in a playground, that goes back and forth, back and forth. So how is that back and forth maintained? Through this fulcrum. Type 2. So the second type of lever is where the fulcrum, so this is the fulcrum, so that is located at one end, and the effort input or the is at the other end. The third type of lever is where the fulcrum is located at one end, and the load and the output load is located on the other hand. So here you can see the fulcrum is located in the middle. In the third type, this is located in the front or the back, where the input and this output load is located at the other end. Now, the importance of this placement of fulcrum and this load is because of magnification. Now, I want you to again go back and read because the importance of magnification, because the length of the load arm, so the length of this load arm, the length from load to this fulcrum, the length from the fulcrum to this input arm, or the length of the effort, this determines the magnification. Why? Because whatever response is recorded, it will be magnified, right? Now, the magnification should be as such that it is within this chymograph drum and not, going out. The response does not go or move out beyond the recording graph. So that's why the magnification, it's based on the mechanism which amplifies the contraction. It depends on, this magnification depends on the type of tissue used. So if it's a slow contractile muscle that is used, then the magnification required is 10 to 15 times. If you're using a fast contracting tissue, then the magnification required is 5 to 10 times. So again, detail of all this is given in the book, which you should go and read to understand it better. So I just wanted to give you a brief overview of the assembly, the parts, its importance. Now we come to most important part here, as I kept on mentioning, physiological salt solution. Now each type of the salt solution, like, it's very important to maintain tissue outside the animal body, right? And it fulfills their internal environment and ions and nutrition. So that is the importance of physiological salt solution. It is also called a Swinger solution. Now, it is always important to select PSS in which tissue lasts the longest. The components of this PFS is very important because each component has its own peculiarity. So when there is sodium involved here, the major extracellular cation, the importance of it is it maintains osmolarity and it makes the solution isotonic. Potassium, it's a major intracellular cation. So importance of this is, in the nerve conduction and muscle contraction. Magnesium, why is it used? It is the second most common intracellular cation. It reduces spontaneous contraction of the tissue. Third most important here is calcium chloride, important for contraction and relaxation coupling. And it maintains the integrity and permeability of the cells. Next is bicarbonate and sodium hydrogen phosphate. This acts as buffer. And and the last most important thing is glucose. Glucose is the major nutrient for the tissue. Now, most important here is, first you need to know the components, its importance of it, and why is it used, right? Apart from that, they can ask you the significance of this main components, which I mentioned here. Now, there are precautions that needs to be taken in during PSS preparation. So one is these precautions that it should be prepared in distilled water or double distilled water. Why? To avoid salting out of dissolved components. Second, calcium chloride should be added last, or so, to prevent precipitation and chelation of bicarbonate, which makes the solution turbid and change the pH of the solution. Most important thing, aeration is important for PSS. So as you had, if you can recall the diagram which I had shown and also the video, in the inner organ bath, when the tissue is mounted, PSS is also added. But there we also have, through that tissue holder, there is aeration where oxygen is provided, right? So aeration is important for PSS. Why? It's done using carbogen. Now, what is carbogen? It's 95% oxygen and 5% carbon dioxide. Another important fiber question. So pure oxygen may interact with HCO3 and PSS, which causes carbon dioxide loss, and PSS becomes alkaline, and therefore the combination of using carbogen, 95% oxygen and 5% CO2 is of importance. Another thing, fresh solution is always preferred in PSS, and it can be stored up to 24 hours using refrigeration. Now remember, when you are trying to store it, calcium and glucose should be added at the time of experiment and not before. Why? Because glucose, it's food, it has nutrients, it has sugar, so it can create bacteria inside. Bacteria can grow. So that's why if you are trying to refrigerate it for 24 hours and use it in the for later or next day, then avoid adding calcium and glucose. Again, I told you the importance of calcium and why it should be added last. This all and you can read through it. Now, again, the most important part, types of tissue. First and foremost, when you're trying to conduct an experiment, which type of animal you're going to use is of importance. That again, you will have to go through the experimental pharmacology book and read it. From choosing the type of animal, why are you choosing that type of animal is because of the type of tissue that animal has. Now, the tissue is of two types according to the tissue of muscle obtained. It can be smooth muscle preparation, skeletal muscle, or cardiac muscle. Or if you are selecting the tissue according to the response obtained by the tissue or the muscle, in the sense, slow contractile tissue or muscle or fast contractile tissue muscle. So the slow contracting tissue muscle, example is frog rectus abdominis, stomach fundus, guinea pig ileum, or tracheal smooth muscle. So these are the examples of slow contractile tissue, and this is another viva question for you. Fast contractile tissue or muscle. So examples, guinea pig ileum, rat uterus, ascending and descending colon. Now, again, depending on the type of tissue you select, it also depends on which type of solution is used. So for guinea pig ileum, thyroid solution is used. For rat colon or uterus, Dijon solution is used. So for rat stomach fundus, Krebs solution is used. So, now you can try out everything, why the importance of PSS, why is it used, which type of PSS is selected, the type of tissue, how is it selected according to the muscle or according to the response. Now, if it's according to the response, is it slow contractile or fast contractile? Now, another most important thing is when you are trying to cut the tissue, depending on slow contractile or fast contractile, those tissues can be cut into longitudinal or circular. In the sense, the tissue will have fibers. Those fibers will have receptors attached to it, right? So a longitudinal muscle will have a longitudinal fiber. So those fibers will, those receptors, they will flow in this longitudinal manner. A so that this is how, when you cut the tissue in a longitudinal manner. Now the tissue that you are cutting through in a horizontal manner, they will have circular fibers. These circular fibers will have the receptors flowing in a circular manner, right? Now suppose whichever tissue you are selecting, it has circular fibers. That means that the receptor is flowing in a circular manner. But you are cutting that tissue in a vertical manner. So when you cut it here, the tissue will not give you the response because the receptors they are flowing in the circular manner, but you have cut that fiber into a vertical manner. So the response will not be there. If I cut this tissue into a horizontal manner, so the circular fibers, they will be cut into a horizontal manner, but the response will still be there. The receptors will still give you the response because they are flowing in the circular manner. Same way in the longitudinal muscle, if I cut the tissue in a horizontal manner, the receptors will still give me the response because they are flowing in this way. But if I cut it in the vertical manner, they will not give me the response because I've cut the flow of the response of the receptor that will respond. So I hope I've made myself very clear, in the importance of the tissue that you select and whether it's slow contractile or fast contractile. How you cut the tissue is also important because of the circular and longitudinal fibers and the response that they'll give you. So with this basic understanding, now we come, I know we only have 10 minutes, so I'll try to do my best. But during your practical examination, how do you present an exercise during practical examination? So suppose it says first, the question is with you're given the question paper, the examiner comes to you. So even if you're using a simulation model, how will you present? So I'll say, good morning, sir. The exercise given to me is such that with the help of the simulation model, the model that I'm using is CalPharm version 2.0. I will demonstrate the concept of competitive antagonist of histamine by using isolated smooth muscle preparation. So again, how would I present this exercise? Good morning, sir or ma'am. The exercise given to me is this. Using the simulation model here is XFarm version 2.01. Here I have selected guinea pig ileum as the smooth muscle preparation and Krebs solution as the physiological salt solution. With the temperature of 30 to 35 degrees Celsius and pH of 7.3 to 7.4. After selecting the appropriate tissue and PSS with a suitable temperature, I have recorded the baseline, followed by which the lowest concentration of histamine is added to the inner organ bath and recorded the response. So even if you're doing a simulation model, but this is how you need to present to the examiner, so they exactly know that given the exercise, which type of model you have selected, if it's competitive antagonist histamine, so why you chose guinea pig ileum, and the scraps, the PSS, the temperature, everything, the muscle preparation. Here I've selected smooth muscle preparation. So everything, the examiner will automatically know that you know your thing. And as I mentioned before also, whatever you say is your next Viva question will lead on to that, right? So this is how you present an exercise during practical, whether you are actually performing it or whether you're using a simulation model. You also mention the simulation model and the version number that you are using, right? Now, what I'll present here today to show you is the second exercise. Find out the concentration of histamine in the given solution by a 3-point bioassay. So, which is also known as 2+1 bioassay. Now the experimental procedure examples, and I'll also show you the recordings which I had done. I was fortunate enough to attend one of the animal handling workshop where I was able to, record the demo that we had done over there. So the first and foremost, whenever you perform an experiment or even when you're reading about it, comes the dissection part. And I'm just telling you the steps of this experimental procedure, because with every step, as I told you, there will be Viva questions to it. So the first and foremost is using the experiment which I showed you previously. So here I'll prepare the thyroid solution in distilled or deionized water. The first question, why distilled or deionized water? Why not a normal saline? Why not anything else? So, using a distilled or deionized water for thyroid solution, it is to avoid the interference of unwanted ions, to ensure accurate ionic composition, to maintain the physiological pH and osmolarity, to keep the tissue viable, and thyroid, it mimics the extracellular fluid. Now, atropine is added to the thyroid solution. Why? Why is atropine added to the thyroid solution? Because atropine is a muscarinic receptor antagonist. It blocks the parasympathetic effects. It prevents spontaneous acetylcholine mediated contraction. And it ensures responses are due to the test drug only and nothing else. That's why I told you it is important for you to know, according to the experiment, which type of tissue PSS you are using, and why are you adding atropine to that thyroid solution. Next, we come that guinea pig is kept overnight fasting. So I had mentioned the animal I'm using is guinea pig and the tissue I'm using is guinea pig ileum. So guinea pig is kept overnight fasting. Why? Because to empty the intestinal contents, to make the dissection easy, to prevent contamination, to reduce tissue damage. If the any other human patients also, if they are undergoing surgery, they are kept meal by mouth 24 hours prior. Why? Because we don't want the food contaminant to get disrupted during the surgery or to regurgitate. The patient's stomach can regurgitate during the surgery. So the same way, guinea pig is also kept fasting overnight so that we prevent such kind of contamination, regurgitation, or tissue damage. Then comes the animal is sacrificed by stunning. The moment you talk about stunning or by head blow, then the Viva question will come on euthanasia. Then neck vessels are cut and the animal is bled out. Abdomen is opened through a midline incision. Ileocecal junction is exposed. The terminal ileum is cut after discarding 10 centimeters nearest to the ileocecal junction. Now, as I told, anything that you speak and whatever you speak, a question will always follow. Why? How? Why? How? So here, first, you're opening through the midline incision, ileocecal junction exposed. How? So the area near the junction is thicker and it's less uniform. It may contain, spear patches or something like that. So there is variability and variable sensitivity to the drug. So for consistent experimental response, that junction is exposed. Now, how is it exposed? By cutting, the terminal ileum is cut after discarding the 10 centimeter nearest to the ileocecal junction. So why discarding the 10 centimeter nearest? Because there can be sensitivity of the drugs. There are various receptors. It's not that we've learned about receptors, right? So there would be other receptors also. So when cutting near that segment, other receptors may give you the response which you don't want. Therefore, we are cutting that part of the tissue and discarding it. Now, the mesentery is cut close to the guts to prevent the injury of the intestinal wall, to avoid bleeding, and to maintain the tissue and the tissue integrity. So, as I told you, whenever you speak, even in this dissection part, each procedure, each step will have a why or a how question, and your viva will go accordingly. So the mesenteric attachment is cut as close as to the gut as possible without injury for a distance to about 20 to 25 centimeter. Moving forward, the intestine is then cut across the lumen of the isolated piece through the lumen of the isolated piece is thoroughly cleaned by running warm thyroid solution repeatedly through the proximal opening of the help of 50 ml volumetric pipette. I'll show you a clip so that you'll get a better understanding of it. Now, what you need to remember is, there should be undue stretching, ballooning, or handling of the gut. That all should be avoided. And the clean strip of the intestine, once that tissue is cut, the clean strip of the intestine is then placed in the fresh, warm salt of thyroid solution for a short period before being put up. Now, why? It's very important to give that tissue a rest first. Second, you put it in a fresh, warm salt thyroid solution. Because you've cut the tissue, now you're taking that tissue out of the body, right? So the temperature shifts. Now you want to balance that temperature. You want to give it, as I mentioned, in vivo, in vitro, in silico, the definitions. So you're cutting that tissue which was maintained at a body temperature inside that kinetic ileum. Inside that kinepig, you're cutting that ileum out, bringing out in the external environment, but you want to maintain the temperature as it was inside the body. So therefore, you're putting it in the fresh, warm thyroid solution. At the same time, when you're cutting the tissue, bringing it out, the tissue is under stress. So you wanted to let it rest for a short period of time so that, to avoid the spontaneous response once it's mounted. It should be well rested enough. Now, if the strips are kept for further use, they are better placed directly in the ice cold aerated solution and kept in the refrigerator overnight. So again, viva questions on PSS. I had told you, if you are making the PSS solution, but now you are using it for later, then what precautions must be there to preserve it. So here again, I had mentioned to you about the longitudinal circular fibers, and I'll just play this clip for you all to see. Okay, why is this clip not playing now? Okay, there you go. So I hope the video is visible to you. So this is a petri dish, as you can see. Then this is the aeration tube where the tissue is mounted here, right? This is a guinea pig ileum. Can you see how the tissue is cut, right? So it's in this square manner, and the person who is there, he's trying to dissect the extra skin out. So with a forceps. Now you also need to remember that you don't take a toothed forceps, but you take a blunt forceps to avoid injury to the tissue. And he's trying to take out the extra layers. If you can see clearly or observe here, you can see this is called the lumen. This is where you'll tie the tissue and mount it to this tissue holder. They are giving oxygen aeration here, but as you can see, the aeration, it's so fast that the PSS is spilling out. So because of that, now he took a tuberculin syringe. He took some of the PSS out so that he can now put it inside this lumen. So he's now clearing or cleaning this strip of guinea pig ileum. He's putting the PSS inside this lumen to clean the strip, and also that it runs through the PSS solution. So this is what I wanted to show you, how the tissue is cut. Again, if you can observe, this epithelium will have circular fibers. Hence, it was cut in this rectangular manner. And the person conducting this experiment is putting physiological salt solution through this lumen to clean it. So I hope you can see this, and with that, move on to the next slide. Again, you can clearly see. So he is holding the tissue with a blunt forceps and removing this extra layer of fat, extra layer of tissues that we don't want. And you can see the lumen here, right? So with this, a small segment, usually 4 to 5 centimeter of length, depending on the organ bath, is cut. A small thread is passed through the lumen and the wall near the mesenteric attachment at each end with the help of the needle. This also I'll show you a clip. Now one end of the segment is tied securely to the tissue holder and transferred to the organ bath, which is already filled with the thyroid solution and bubbling with the gas. Now hold both the tissue holder and the long thread in the same hand. Why? Because if I'm holding, this is the tissue holder and this is the thread. If I mounted the tissue here, tied the thread. If I'm holding it with two different hands, there are chances while I'm taking it out and mounting it in the inner organ bath, I'll stretch the tissue. That's why it's recommended that you carry both the tissue and the thread in the same arm to avoid undue stretching of the tissue. So the tissue holder is fixed in position with clamps, and the long thread from the tissue is fixed in a frontal lever, adjusted suitably for tension and magnification. I'll show that clip also. So here you can see now the tissue, one side of the thread is okay. So one side of the thread is tied to the tissue, and then it's tied to the tissue holder, and this is the second thread which will tie. Now please remember, when you are tying the thread to the tissue, one side you're tying it to the proximal end, the second end you're tying to the distal end. But make sure that the thread is tied in a parallel manner and not in a diagonal manner. Because this is when you mount the tissue, it should lie in a straight, like it should be in the straight manner. The tissue should be in the straight manner, right? If you tie the proximal end here, and then if you tie the proximal end here, and then you tie the distal end here, the tissue will bend. That's why it's important when you tie the tissue, the proximal and distal end should be tied in the same side from the lumen. And also the proximal and the distal, the lumen should not be tied together. You will block the lumen if you tie one thread here and the second thread here. You want to tie one thread here and then in the proximal part, and then the second thread right opposite to it in the distal part. So I'll just play this clip so you can see here. So he took the needle, now trying to tie the thread. One thread is tied in the proximal end of the tissue, which is tied to the tissue holder. Now he's trying to tie the distal end. So he's putting the needle in the distal end. Mind you, if you can see, the proximal and distal are in the same parallel lane. Very gently he's trying to tie the needle. The thread is passed. You have to be very gentle. Can you see? The same line. Now can you see how the tissue is stretched? So you have to handle it very carefully. Very carefully. And someone else is also pouring PSLs just to maintain the temperature. Okay. Now one more thing, as you can see this. So this is so ideally, oxygen is given. So oxygen or aeration is given here in this tube. But they are giving it through this. If you can recall, an aquarium, they also have air bubbles inside. When you can see the fishes are floating, there's air bubbles are there. You know those. So this is a oxygen machine that is found in an aquarium. So in such cases, when you don't have an oxygen cylinder to properly provide the oxygen, then artificial aeration can be done through using this pump, oxygen pump, from an aquarium. So you can see tissue is tied to the tissue holder. The thread is tied in the proximal end, and now he is tying the thread in the distal end. Again, in the same line. You have to be very careful while you're trying. It seems easy, but the tissue is so sensitive, you can easily break it if you stretch it more. Now he'll cut the thread, and this is the distal part. The distal part, you don't need in much thread, right? Now this is what I'm telling you. Hold the tissue holder and the thread in one hand instead of two hands, as this person did, because there are chances while you're trying to put it inside the inner organ bar, you will stretch this tissue and it will tear apart. So now he's putting it in the inner organ bar. Ideally, the inner organ bar should be started with aeration. You can see the fulcrum, the lever is already set. The load to keep it straight, the magnification is set. Now they're mounting it. They've tied it. Now this thread will be tied to the liver, so that once it starts contracting, the response can be measured here. But once the mounting is done, the tissue should be rested here for at least 30 minutes, because again, I told you, it's gone through a lot of stress. It needs to, it needs to be rested well enough so that we don't have spontaneous contraction. Another most important thing, if you can see, the aeration, the bubbling, it is so fast. It's so fast that the tissue is, moving back and forth. It's touching the inner organ bar. And we have to avoid that. You see, it's moving so fast, it's touching the inner organ bar. So you need to remember the speed of aeration that you are giving, right? The oxygen. So as you can see, the oxygen pipe is attached to this tissue holder or the aeration tube, and it's given through that machine which I showed you. They did not have an oxygen cylinder, so that is how they are using to give aeration. The tissue is mounted to the tissue holder. The thread is then tied to the liver. It's rested for 30 minutes, so to avoid spontaneous contraction, and then the response will be taken. So again, this is what I'm trying to tell you. You see how the tissue is moving so rapidly? We should avoid that. Once it's well balanced, you see the response. Spontaneous response is showing. This we should avoid. We should give the tissue for at least 30 minutes. But you see, how the liver is moving itself. The tissue is responding. It's contracting. So frontal liver again. Viva questions on the types of frontal liver, the types of liver. It's a type 1 isotonic liver. So isotonic, all that can be Viva questions. Now, before tying the tissue to the frontal liver, the liver should be balanced horizontally with equivalent weight of the tissue in this 0.5 grams of plasticine. This is also a Viva question. At the point of attachment of the thread. Now, after balancing the liver horizontally, tie the thread of the tissue to the liver at the place of 0.5 gram weight and remove the weight. Then keep the weight or the plasticine on the liver between the fulcrum and the writing point for 30 minutes, which I told you why. So this is kept there till the tissue is rested. So you can see, we are trying to balance the liver. So we've kept a load here. We've kept a small load here. Now once so that it's in the horizontal manner and the tissue is rested. Once this tissue is rested, we will lift up this load and put it here, so that in order for the tissue to give us the response. Otherwise, if the load is equal in both the ends and it's balanced, the tissue will not be able to give us the response, right? So this load should be lifted. Now the procedure. So about 10 gram of histamine is weighed and dissolved in 10 ml of distilled solution. This is very important. I'll go through this step and then I'll make you understand the stock solution. What is the stock solution? What is the maximum concentration? What is called as the minimal concentration? So about 10 gram of histamine is weighed and dissolved in this 10 ml of distilled water. So this 10 gram dissolved in 10 ml has the maximum concentration, maximum known standard concentration. And this is what is called as stock solution. Stock solution is the solution which has the standard known concentration, the maximum concentration, right? From this stock solution, we will start doing serial dilution in the descending order. So this contains 1000 microgram per ml solution, and 1 ml of this solution is taken, mixed. So 1 ml from this 10 ml, which has the maximum concentration, which is known as the stock solution. 1 ml is taken out and mixed with 9 ml of distilled water. This contains 10 microgram per ml. So I told you the importance of why we are using distilled water also. Now 1 ml of the above solution is again mixed with the 9 ml of distilled solution here. So from the stock solution, which has maximum concentration, we take 1 ml out, dissolve it in 9 ml of distilled water. We take 1 ml solution from here, dissolve it in the next 9 ml of distilled solution. So after 30 minutes of relaxation of this strip of guinea pig ileum, the tissue is washed for 2 times by filling and emptying the inner bath with the titrate solution, and then we choose the concentration. So this is how a serial dilution is done. And the last one, which it will have the minimal concentration. So the first test tube which has the stock solution has the maximum concentration and will keep on diluting. The last test tube will have the minimal concentration. So these are the Viva questions. Now the drum is started, baseline is recorded for 30 seconds. And 0.1 microgram of histamine is added gently into the inner bath, and drum is switched on 1 revolution per 96 seconds. Now, since the guinea pig ileum is highly sensitive, tissue contracts immediately and liver draws the response. After 50 seconds, the drum is stopped, the tissue is washed. Immediately, the tissue relaxes and liver comes down to baseline. Again, another wash is given to the tissue. Again, switch the drum and allow it to run for 30 seconds. Why? So, the key thing here for you to remember to know is the dose cycle and the contact time. The dose cycle, it's the time gap between drug dose additions while plotting the dose response curve. So it usually takes 3 minutes for a fast contracting tissue or 5 minutes for a slow contracting tissue. So that's why again I told you the type of tissue you select is very important, because the response will be accordingly. Baseline dose response and then again the baseline. Different for a fast contracting tissue, different for a slow contracting tissue. Now contact time means the time duration during which the tissue comes in contact with the drug. Now, one thing I want you to find out after this session ends is what is biophase. So, as I mentioned, the contact time of 15 seconds and 2 times washing of tissue should be maintained constant throughout the experiment. And dose selection is also very important. So slow contracting tissue will have a different baseline dose response, and again the baseline. From one baseline to the end of the dose response is known as the dose cycle. Between the two baseline and the end of this dose response is known as the contact time, which will differ for a slow and fast contractile tissue. This is something just to show you. One, that we did not have an oxygen cylinder, so they use this oxygen machine from an aquarium. So this is one of the questions that can be asked. Second thing, here usually we smoke the paper, but if we don't have that time to smoke the paper, then you can use a white paper. You can use a marker at the tip. The tip of the liver is known as style. You can put a cotton bud with an ink. You can see here we have an ink here. Or we can use a marker also, to the tip of the marker attached to the stylus, which will take the recording of this. So these are very common Viva questions that can come up. If you can't smoke the paper, what can we do? If the light runs out, how will you maintain the temperature? If you don't have the oxygen cylinder, how will you give aeration? What is the frequency of giving aeration? What is the importance of giving aeration? See, I told you it's 351 here, and there's so much to tell you, and I can't cover everything, but this is something for you to go back and read. Everything can be a Viva question. So here I already demonstrated the bioassay assembly. This was one which we had set up in during our residency for the final exam. And as you can see, the paper here that was smoked and the liver. So when we do bioassay, there are always advantage, disadvantage, and there are always errors. We are all human beings. No one is perfect. Error is there. So there are two different types of errors that can occur during bioassay. One is biological, second is methodological. So down regulation of receptors due to repeated washing, avoiding overwashing. Second is loss of tissue sensitivity. Animal use of different species, sex, age, weight can give you an error. That's why it's very important to know the animal selection, and you doing the correct animal selection for the experimental you are going to perform. The laboratory conditions, which may be variable, the housing and handling of animals, it should be by qualified experienced staff. So all this anywhere can give you a biological error. And there is methodological error also. There can be human error, there can be experimental error. So with this brief overview of theory as well as practical part, I conclude my session in saying that bioassays are vital for drug testing and potency estimation. They require standardization and careful setup to reduce errors. And the limitation, of course, are ethical issues, more laborious, time-consuming, and skill-dependent. And for that, because of those limitations, we are now, in our MD curriculum, adapting to simulation bioassays, which also has advantage as well as disadvantage. With this, I would like to give you my references of this session which I have shared with you, or which I have discussed with you, using Fundamentals of Experimental Pharmacology by M.N. Ghosh, the 7th edition. I highly recommend everyone to read